Good morning, everyone, and welcome to this first internal webinar of OBSGESSION. So before starting, I just ask to inform all of you that this webinar will be recorded and also transcribed. So the presentation parts will be eventually be available to the public, but not the discussion parts. So I hope everyone would agree that we are recording this webinar. So can you see my my presentation, right? I just want to give you a quick overview about the agenda. We have for this webinar 2 presentations. The first one will be presented by our esteemed colleagues at ITC, Andrew Skidmore, about the essential biodiversity variable. And the second presentation will be addressed by the other esteemed colleague, Marcel Buchhorn. I hope the pronunciation would be correct. So, and both presentation each will be for 30 minutes. And after finishing the presentations, we will have 1 hour to have open discussion. So please keep your questions for 1 hour to yourself, and then we can discuss and you can ask your presentations. So I stop my sharing now, and then I give a floor to Andrew, and if he share his presentation. Andrew, floor is yours.
Thank you, Elnaz. Good morning, everyone. I hope this PowerPoint comes through okay now. It's saying it's loading it.
Yeah, we can see it.
You can see? Oh, perfect. Okay, great. Well, yeah, we thought we'd kick off this first seminar with a discussion and talk about data and what we're going to call the different data and the different layers that we're using in OBSGESSION project. And I think this is quite important because we're already seeing a lot of terms being used by colleagues and, you know, calling things essential biodiversity variables and calling other layers you know, biodiversity information and so on, and it gets a little confusing because there is I think quite a useful formal structure now has emerged around the use of these terms. So we'll try to this morning go through these quickly and try to get that straightened out. So at the end of this talk, I hope everyone is at least familiar with the terminology and also what data and is available via VITO. So that's the basic aim of this session. Okay, so essential biodiversity variables. I thought it would be best to start with a discussion where it all started, actually, which is essential climate variables, or ECVs. And these have been around for a long time. The global GCOS, the Global Change Organizing, no, Global Change, I can't even see it, what the acronym means there, but basically it's the Global Climate Observation System. And they quite early on realized, maybe 25 years ago, that they had to come up with some metrics to measure climate. And they had it, I think, fairly easy relative to the biodiversity world because they quite quickly were able to generate something like you see in this graph, which was temperature anomaly and showing the global mean estimate of temperature rise. And that meant that you got quite a dramatic graph, which people could just say, oh yeah, temperature is rising. And so you got one view of a dynamic which sort of meant something both politically and socially and in companies. And they went further with this, and by now they've got more than 50 essential climate variables have been defined, and they work across atmosphere, ocean, and terrestrial. They've been defined, what they call essential climate variables. What's characteristic of them all is that they're basically all abiotic. Virtually all of them are abiotic, not all, but most. And most are quite easy to measure in terms of actually, just saying, okay, I'm going to the field or I'm setting up an observation and I want to measure whatever it is, temperature or water vapor or so on. It's got very clear ways to measure. If we look at particularly the terrestrial side of this, what we see is that the terrestrial are again largely abiotic in terms of the variables that were mentioned, but you also have a number of biotic, including, say, land cover, vegetation type, FA par, leaf area index, above ground biomass, and so on. So it was mostly abiotic, but there were a few biotic or biologically orientated variables in this list of ECVs. So what about these EBVs, or essential biodiversity variables? Well, it's pretty obvious to everyone that the global biogeosystems have been altering, and humans are impacting the Earth's ecosystems and biology. And one of the interesting things is that there's even been a discussion in the last 2 or 3 years, and that there may be an anthropocentric signal as a new geological like Pleistocene or so on, and they're calling it the Anthropocene. And the idea is that there's actually humans are altering the landscape of the Earth so much that this will become impacted into the geological record. So, in a few million years' time, people would be able to see, oh yes, there was definitely an impact of humans. But this has actually got a very long-term view on it as well, EBVs. But we're basically looking, how can we monitor change with these essential biodiversity variables? Now, this little movie I made up, this is Opera House in Sydney. And you see that at the Opera House, you've got this reef running around the outside of it, and it's got about 150 species there. But most of it is no biodiversity at all. If you look at the Opera House area, but also the centre of Sydney, you see it's all, typical city. So very extreme conditions for any biodiversity to exist. You see a few plane trees and pigeons and rats and that sort of thing, but nothing much else. But right next door to the CBD, you've got the Botanic Gardens. And here you've got 50 different plant species per hectare, with a total of about 9,000 plant species over 180 hectares. And a bunch of native animals have also managed to come into this area. So you see there's very different amounts of biodiversity, even in a city like Sydney. You've got marine habitats. There's about 3,000 species have been observed in the Sydney Harbour. Finally, you can go to an area of sort of remnant bushland. This is Bradley's Head National Park. And here there are around about 100 plant species per hectare in natural forest ecosystem. And just by way of comparison, if you look at a typical tropical rainforest, you may have 150 to 200 plant species. So these dry sclerophyll forests are a bit of an anomaly from a biodiversity perspective, because you've got incredibly high biodiversity in very poor soils in a sort of a temperate environment. So you've got the areas in there's areas in South Africa which are similar to this, and also in France, where you've got some quite poor country, but soil-wise low nutrient, but you actually have very, very high biodiversity. So even in a couple of kilometers in a city of the size of Sydney, you've got these incredible diversity of species occurring. And rather the ECVs were as I said, quite simple, that there's a number, but the problem that's always been people have struggled with biodiversity, of course, is that it's more than just a number. There's, how do you define biodiversity? The first term, the term was actually proposed by Dasman in '68, and there was this first definition adopted by the Convention on Biodiversity in '92, and they were talking about diversity within species, between species, and ecosystems. Since then, of course, we've recognized that there's quite an important genetic or phylogenetic component in biodiversity, and so that's been added in the form of essentially genetics. Nowadays, biodiversity is being recognized at 3 levels: genetic diversity, which is the genetic variation contained in the genes of all species; the species diversity, which is the number of species and the number of individuals per species; and then ecosystem diversity, the variety of habitats, communities, and ecological processes. So we've got this, as I said, we've got these typical things: species diversity and habitat, which we're all very familiar with. And probably the area that's developing most quickly at the moment, in one of the most exciting areas, is genetics, and how genetics can be used to identify variety, but also to look at how the tree of life has expanded. And I think this is one of the coolest graphics that I've ever found, which is showing the tree of life from the first Earth, and showing how different branches of biological life evolved over time. And right at the end, of course, we've got humans, which were very late, of course, appearing, but this is nothing new for any of the people on this call. There's ecosystem services, and yeah, these are just some of the estimates of the value of ecosystems and biodiversity: water retention, flood control, soil quality, pest and disease control, genetic variability, pollination, and so on. So there is, quite substantial estimates people have come up with, scientists have come up with, about the value of ecosystem services. So the challenge that emerged was very much, is there a single indicator like temperature increase that could be used to show biodiversity change? And the Global Living Planet Index, which the WWF started in the 70s, they've shown, using a technique based on data that was actually collected from observations. So these are observations which have been verified, either government or published data sets that can be verified. And using these data, the Global Living Planet Index has been tracking how species have been declining, and this index shows a decline of 70% by now in the last 50 years. And people have actually thrown criticisms at this. It's, based on how do you weight it, and the techniques, the modeling techniques that are being used over time have changed. There's also problems with scalability and comparison, but at least it is a single number. So a lot of people see this, it's like temperature, and say, oh wow, that's amazing, that you can see this long-term trend. More recently, there've been Living Planet Index was mixed up with the use of the IUCN estimates of extinctions and declines in populations. So typically they were saying, with the Living Planet Index, what was happening in terms of declines. Now we're looking at this combination of all the different IUCN categories, and this is a typical kind of study which is now being done, where we can see that there is a global decline, as we said, about 70%. But we can also start to look at how that decline is happening over time by different regions. And you get some very, very different patterns emerging from this Living Planet Index when you look at it by region and time. Yeah, well, you see globally the decline is fairly constant. The darker greys represent greater declines. But you can see things like Europe actually had declined, and then it actually improved again, and then it declined, as an example. The Asia Pacific and Africa are simply just going backwards, as is Latin America and the Caribbean. So you start to get quite different, using these type of data, you can start to get different views of what's happening, both temporally and regionally, spatially, around the globe. So what's been the response to this? Well, the international response has been, firstly, the UN Sustainable Development Goals, the SDGs, and also the CBD, Conventional Biodiversity Aichi targets. And these two actually map into each other. The UN Sustainable Development Goals go much more broadly, as you know, than biodiversity, but the CBD really has been focusing on biodiversity indicators. And since the 2020, the post-2020, so Aichi went from 2010 to 2020, the CBD Kunming Montreal post-2020 biodiversity targets, they have been reworked, and we've now got an updated framework of themes and targets to work with. But we still have this problem, actually, how to define and how to measure biodiversity. And the definition of EBVs was basically came out of this paper by Pereira and colleagues in 2011, where they were looking as EBVs as the derived measurements required to study, report, and manage biodiversity change, focusing on status and trend in elements of biodiversity. And how you do that? Well, there's not really a definitive sort of list of these essential biodiversity variables, but what we'll show now is that it's beginning to stabilize around a few EBVs. So some of the key words here are that they should be essential, they should be biological, they should be policy relevant and feasible. So you should be able to do them, right, calculate them. Ideally, they should be scalable. So an example of a scalable EBV would be something like leaf area index. You can measure leaf area index at like 1 centimetre, and you can also measure the leaf area index of the globe. So it's scalable across, huge ranges. They're the ideal ones, especially to calculate and use. They should be ecosystem agnostic. That is, ideally, it should work everywhere, whether you're on an ocean or freshwater or terrestrial. So what would be an example of that? Something like chlorophyll would be something that's ecosystem agnostic. You can measure that biological variable across all these. And it should be able to be looking at change. How are things changing so we can monitor it? Now, this is the original figure here from Pereira's paper. And what you see is that you've got these primary observations of change in biodiversity, which is the in situ, the point-based model at points, and the remote sensing. And then you come to the essential biodiversity variables. And what's important here is that there are these 6 what are called EBV classes: genetic composition, species population, species traits, community composition, ecosystem structure, and ecosystem function. And what's interesting is since 2011, these EBV classes have been stable. They haven't changed. The EBVs themselves have changed, but the EBV classes have been very stable, which indicates to me that actually they pretty much nailed it in that paper about the broad classes of how to categorize. And then what Pereira and colleagues suggested was to take the EBVs and come up with high-level indicators of biodiversity and services, and then use those in turn for building scenarios of biodiversity. So we've pretty much, I think, in Obsession adopted this, right? We've got different work packages which are dealing with different elements of this type of process, from the fundamental primary observations through to the EBVs, in this case classes on this figure, and then going into the indicator, high-level indicator, and then the modelling and ecosystem services. What are EBVs? Well, essential biodiversity variables. You've got these EBV classes, and then you've got the EBV examples. So within each class, you've got a series of essential biodiversity variables, and then you've got to be able to measure them. You've got to be able to look at how temporally sensitive is it, and can it be measured with a reasonable temporal sensitivity required for the job at hand? Is it feasible? Like, are the data available, or could they be available in the near future? And what's the relevance for policy, say for CBD targets and indicators? So the official GeoBond EBVs and EBV classes. So here we've got the 6 classes: genetic composition, species populations, species traits, community composition, ecosystem functioning, and ecosystem and structure. So by now, if you look at the GeoBond site, it's pretty much stabilized into a number of different EBVs. So you've got, let's say, genetic composition, it's got genetic diversity, genetic differentiation, effective population size, and inbreeding. So for species population, you've got 2 EBVs, species distribution and species abundances. So it's quite a clean and simple system with an EBV and an EBV class. The problem is that as scientists, we're now inventing lots of additional data, which everyone is calling EBV, right? So you come up with your favorite product, and then people are saying, oh, that's an EBV, that's an essential biodiversity variable. And that's where it gets really confusing, because according to the GeoBond website, these are the official EBVs and their names, and yet we're seeing lots of people in the literature also publishing, oh, we've got these different EBVs. It gets even more confusing, unfortunately, in Europe, because Europe has gone ahead in EuropaBond and talking about EBVs across different domains of the global ecosystems. But more confusingly is that they've actually identified 84 EBV candidates. So rather than having, let me go back with the EBV, we've got the 4, 6, whatever, sort of 20 or so EBVs, right? Which is the number officially in GeoBond, the Group on Earth Observation Biodiversity Observing Network. Here we've got 84 proposed by Europe. And you start to see, for instance, in the terrestrial realm, that they've taken terms like species abundance and species distribution, which are the classic normal GeoBond terms, but putting in all sorts of additional terminology of terrestrial mammals and of lichens and all sorts of things, right? And then, so basically you're starting to get a system here which is very different and diverges quite strongly from the system in that's proposed by the GeoBond. Not a good thing. Marine, same story. Freshwater, same story. Lots and lots of EBVs are being invested. So we're proposing that Obsession uses the official GeoBond EBVs to be globally consistent with colleagues around the world, but we can discuss this. So the various classes, your genetic composition of EBVs, again, I don't really have time to go through this, but you've got, as I said, they've got these 4 genetic compositions. Now, each of these EBVs now have got a paper associated with it, and each of the EBV classes. So genetic composition, species population has got a good publication that's defined species populations and gives examples. Species traits, ditto, a paper on this community composition, another paper describing it, ecosystem function and ecosystem structure. So the 6 EBV classes have got now have got well-defined EBVs through GeoBond, and they've got good solid articles that have been written describing each EBV class. Now, the problem we struck quite quickly was that we have to prioritize these remote sensing biodiversity products. Why? Well, very simply put, we have two phenomena going on. First of all, a lot of remote sensing derived biodiversity products people have been calling EBVs, and this is causing mass chaos and confusion. You know, a remote sensing biodiversity product is a product. It's not an EBV. And the second problem was that the funding agencies and companies and the organizations like NASA and ESA, the space agencies, they're saying, where should we put our money? What sort of variables should we be prioritizing in terms of products should we be prioritizing in terms of usefulness? So there's a big call from users to come up with a list of what should we be prioritizing in terms of the products that we can generate from the remote sensing. Now, if we go and look into this in a little bit more detail, we see, first of all, we need the remote sensing because there's a lot of taxonomic gaps in the biodiversity. So generally, the biodiversity that we've named so far, named species, the ones we can see, right? So we know that there's about 1.5 million species have been described formally in scientific literature. And the current estimates for the number of species on Earth range from 5.3 million through to 1 trillion based on latest eDNA data. And the widely accepted figure of about 8.7 million species comes from a technique based on the patterns in the number of species at each level of taxonomic trees. So the problem is here that the named species are not necessarily the same as the total number, which could be 1 trillion. That could be 1 trillion species, right? This is, you know, just a huge difference in between these. There's also a lot of spatial gaps in the observation system. If you look at the Living Planet database for 2010, you see the actual data points that WWF were using to come up with the Living Planet database index. That's been filled in by 2020 with more points. But still, you see that there's large areas of the Earth don't have any observations. We don't have a systematic stratified sampling scheme here. We've got overrepresentation of certain regions. We've got overrepresentation of certain groups, biological taxonomic groups. And it's based on the scientific and government literature. So you're not capturing a lot of the information, for instance, from citizen science. So a number of colleagues on this call came together and we brainstormed about trying to identify essential remote sensing biodiversity products. And we basically had a meeting with about 2 meetings with about 40 people on each meeting and reduced the number of essential EBVs and then looked at that within which remote sensing products were relevant. And then we tried to come up with a method to prioritize. And it went something like this, that we had this expert group and there were prioritization criteria, and these are based very much on the essential climate variables. And then using that, we came up with a method to prioritize the ranking within each EBV class and the ranking across all classes. And so this is typical way it worked. If we take, say, something like genetic composition, you've got allelic diversity. And what we said with remote sensing, we can't measure that from space at the moment, right? So there's just no data. If you go to something like species population, you're looking at candidate EBV would be species distribution, and then you could look at something like tropical tree species distribution, which actually can be measured with remote sensing using, say, LiDAR in combination with image spectroscopy, and so on and so forth. So what we found in all were that there were around about 120 remote sensing biodiversity products, and these were prioritized according to these 4 criteria, which were actually used by the Essential Essential Climate Variable community. So we were looking at relevance, feasibility, accuracy, and maturity, and these were all given a score and ranked. So each product was sort of grouped and made, and let me say, standardized into some name, because a lot of people had different names for the same product. That was another confusion, right? So we first of all tried to standardize it into a certain product name, and then we had these prioritization products. And in the end, in this paper that was published in Nature, Ecology and Environment, we've got a priority list of remote sensing products which are important for biodiversity. And here you see the top 30 biodiversity products which were listed in the paper. And what's interesting is that, for instance, what came out most highest priority are things like what we called biological effects of fire disturbance and biological effects of irregular inundation. So they were products which came out most important, both within an EBV class and then across an EBV class. So in all, we did it for 120, but the paper, I think, Eleanor's, from my memory, we listed this 30, wasn't it, in the paper, even though in the appendices, we do about 120. So you can look at each product and how that was prioritized. And yeah, so, this is the way it went. Basically, the expert group looked at something like biological effects of fire disturbance, came up with a ranking factor according to these 4 criteria, and then basically summed that ranking factor across all EBV classes and also within a class. And essentially, we were able to link the products to the SDGs and the Aichi targets in the paper. Now, it could be repeated for the post-2020 Kunming Montreal targets. And a number of these are starting to find their way into Copernicus Global Land Services products that were identified. And now Marcel will be taking it up and talking much more about what these actual products are within the global land services. So just, come back, recap. You've got EBVs, so please stop calling everything an EBV. Call it, you've got about 20 EBVs, which are listed in the GeoBon. I suggest we use that list from GeoBon website in this project. And if you've got products, you can start to see these products are being generated, and some are available globally or Europe-wise through Copernicus, and some are also available from other partners within the consortium. So that's the kind of conclusion, where we've gone through, looked at biodiversity, how much biodiversity is being lost, how do we measure biodiversity, and how can we improve the reporting on the state of biodiversity, and especially using, hopefully we can hone in as a group and use a pretty standardized set of reporting criteria. Okay, thanks. Sorry, I went a bit over there, Elnaz and myself.
It's okay. Thank you, Andrew. Thank you very much for the very nice presentation and thoughtful. I think if you're not tired, and I'm sure that all of you have some questions from Andrew, just keep it after Marcel's presentation. Marcel, floor is yours.
Okay, thank you. I will just start. Okay, I hope you can see my screen. So thank you, Andrew, for your part. And so from our side, we want to talk a little bit how you can find EBV data, biodiversity products, or how you find data to generate them yourself in, for example, models. As you maybe remember, within the project proposals, we already identified EBVs, but as Andrew already said, we have to revise this list because I think this list, what we used in the proposal, is following the GeoBon definition. So we will anyhow revise this list, and mainly what we want to come up is which EBV we can generate within obsessions and what EBV, which remote sensing biodiversity products or biodiversity products we can also produce within the product, which doesn't have to be directly going into one of the EBVs in the end. That is just a visual overview of how the work package or how we will do it in within the obsession project. So we will have, as you can see here, we have remote sensing data, we have remote sensing products, and we have remote sensing biodiversity products, and all of these can go later in data cubes or EBV data cubes and will be available to the public. So within this part of the talk, I want to show you a little bit where or which EBVs can be mainly observed with remote sensing, where you can find these data sets, where you can find biodiversity products, as well as where you can, what other options you have with remote sensing data if you want to go from the raw data. So within the EuropaBon project, there was expert ranking on the questionnaire which EBV classes can be most or best supported by remote sensing. And as you can already see, it is mainly the ecosystem structure and ecosystem functions where remote sensing will have a huge impact in their processing, where for genetic composition, species population, species traits, and community composition, remote sensing is not always the best available data source at the moment to generate them, and they are mainly based on field work and in situ measurements. And within EuropaBon, it was even went deeper. So for each of the European EBVs, or we could call them now here remote sensing biodiversity products, they checked which are the best candidates which can be used remote sensing, and we are coming back again to like things like vertical structure of the vegetation, land cover mapping following the UNIS classification, primary productivity, phenology, like these. If you look in fresh water, again here with remote sensing, mainly you can only see the top part of the vegetation. So mainly you go again with ecosystem distribution or structural complexity in riparian habitats. But there are way less possibilities to use remote sensing in fresh water. And in marine, we even have even more or less possibilities because they are mainly for marine ecosystems. You can use remote sensing for detect algae bloom or phytoplankton productivity. Yeah, we have to find existing EBV data sets. And there are some portals out currently which are listing existing EBVs. But again, be careful, not all of them really following the EBV definition of the GeoBon, so the GeoBon definition. So I will just mark out some of the most important as single slide, but you will have here the links to all of these portals in order to have a look by yourself. So most important at the moment are, for example, the portal, the EBV portal Bon in a Box, which is provided by GeoBon. That is currently under development, no, not under development, but it has to be still filled with way more data. It provides mainly tools and code for biodiversity monitoring. There are some EBV example data sets like species habitat index, species distribution, but they also give a really good overview of biodiversity projects, which are currently running or have been running in the past. So if you go on this web page, you can definitely get ideas where to find additional data from these project web pages. The most important portal, one of the most important portals currently is the EBV data portal. That is definitely for the distribution and visualization of EBV datasets. Currently, as you can see, there are 40, I think there are 40, but that's only domains, but there are over 40 products in. And mainly also the results of the Bonn-in-a-Box pipelines should result to be saved later in this portal. As Andrew already said, more confusion came in when the Europa Bonn project decided to not directly use the EBV definition as provided by GeoBonn, but to specify them in more detail. So they came up with a detailed list of the European EBVs as well as a definition. So I put also the link there where you can find them. That's a GitHub repository, so you can find their detailed definition. But also they came up with a detailed idea how to generate them out of different data streams. So on this portal, you can find and filter data sets, and you can specify them if there should be a product or an integration node or how they are connected to each other. So it's a quite useful web page to also see which data sets are currently available. And these data sets are remote sensing based and in situ based, so they are not solely based on remote sensing. There is a portal which is called Goose Biological Ecological Portal. It is not a strict EBV portal. It's more like an EOV, so an Essential Ocean Variables portal. But a lot of these data sets and EBV are useful in order to develop EBVs for the marine and freshwater environment. So you can also have there a look. There's quite a lot of in situ data sets for mammals, and all other. There is the NextGeo's Data Hub portal, which is also not only showing EBV datasets. There are mostly only currently 6 pure EBV datasets in it, but they have a nice overview of all different data collection based on in situ, remote sensing data, models, and statistics. So if you go there searching on the web page, you can find really a nice overview of detailed data sets you can download and use mainly free of charge. In the next 2 slides, I want to show you mainly where are portals or opportunities to find remote sensing based biodiversity products, which will be mainly input in the generation of EBVs. One of the main important portals are the Copernicus portals. They are also split into land monitoring service, climate change, and marine service, for example. But as already Andrew noted in one of his last slides, the Copernicus Global Land Monitoring Service is one of the biggest data provider on European and global space for biodiversity products. We from Vito have, to be honest, quite a big impact there. We generated a lot of these data sets as well as the pipelines to generate them on a 10 daily basis. So you will find there data sets like LRI, FPAR, land cover, phenology, NPP, GDMP. So all these data sets are generated on as I already said, on 10 daily composites. But mainly they are only existing in 300 meter and 1 kilometer spatial resolution, but for that already starting with the year 2000. And I will show later also why mainly they are not there existing with a higher resolution. But if you go there and you want to have higher resolution data set, they are existing on the Copernicus portal, but mainly starting with the year 2016. A really good data source for remote sensing based biodiversity product is always the Google Earth engine. So you can have a look in the Google Earth engine catalog, which also provides you a lot of data sets which can be used in EBV monitoring or development. By itself, these data sets are for free, but they are not so easy to handle because you have to write your own code to extract them and so on. But I will show a solution for that also later. You have the planetary computer, so that is a Microsoft version of the Google Earth engine, but also there in their catalog, you will find nearly all remote sensing data sets from all kinds of satellite systems you want to love to analyze, but also you find there a lot of computing power in order to generate these data, your own models and algorithms. But the planetary computer, if you want to go in deep with data processing, that is not becoming then free anymore at a certain level. A good data source for any kind of remote sensing and in situ product is the STAC index. STAC is the abbreviation for Statio-Temporal Assets Catalogs. That is, let me say, that's a standard how you can save geospatial information data and how you can catalog it. And these catalogs are directly accessible. So they are not like you have to download the data. Mostly you can directly use them in other platforms like Google Earth Engine or Planetary Computer or the system I will show you also from Vito. This STAC catalog index is nearly all existing data sets which are out there. So it's really detailed, but also here again, it is quite time consuming to go through them. So you really have to know what you are searching a little bit. Now we come to more really focused products like the Terrascope portal, which is provided by Vito. And we are using there the OpenEO standard or the OpenEO platform. Here you can find European products with mainly high resolution. So you will find there's a whole Sentinel-2 database, Sentinel-1 database, Sentinel-2 and Sentinel-1 products. So products at 20-meter resolution, 10-meter resolution. Then we have the Copernicus data space ecosystem, so the CDSE, which is also based on the OpenEO. It is also located here at Vito. So we are the main provider of this. And that Copernicus data space ecosystem provides all Sentinel data or all remote sensing data from Sentinel for whole Europe. So that's the main focusing point if you want to have 10-meter and 20-meter Sentinel data, as well as the products. So there is a huge product catalog. So you will also find there high resolution phenology data, FPAR, land cover, LRI products. They're all available there on demand or for download. So mainly on demand if you're using OpenEO, or if you want to download the data sets from there, you can also do that. And another portal that we also have is Planet. So Planet is a private company. They brought out a lot of planetary, what they call planetary variables, which are in high resolution, 3-meter type mostly, but they're not for free. So they are charging quite a lot. So it doesn't sound a lot, but it's like 25 cents per hectare to only view the data. So you can imagine if you run your test site from several hundred hectares, that can become costly quite fast. But that is one of the biggest data source if you want to have high resolution satellite data, which is with a high frequency. So there you can get really daily observations in high resolution. And I think a lot of you already know the Zenodo platform, which is used mainly by projects as well as by a lot of journals to publish data next to the articles. Also here you can find quite a lot of biodiversity products which are directly usable. And a good additional overview is the biodiversity or the spatialagent.org webpage I brought. There you can find an overview of nearly all portals which dealing with biodiversity data. Okay, do we come to the Earth observation overview, so satellite remote sensing. Currently, we have more than 300 satellites for in the space for operational and research purposes. So you can imagine that there's quite a lot of data sources available and data sets. Every satellite mission can have one to many instruments, so you get even more data possibilities. There's a really nice overview of all past, current, future satellite missions, which is provided by the EO portal. And also our colleagues from ETC have a really impressive catalog of all satellites and sensors, including the specifications of every sensor on their webpage. So there you can even look, for example, for the Landsat, which sensor it is, what are the bandwidths, what is the spatial resolution, when it was existing, and so on. So there you can get a nice overview. But the most important satellite missions, which we can use in OBSGESSION, I will present on the next slide. So mostly what you can see here is a nice overview of all ESA developed Earth observation missions in space, as well as which will pop up in the next years. Most important for most of you, you know already, is Sentinel-1 and Sentinel-2, but you also have in this list Sentinel-3, which is a successor of the Proba-V and Spot-VGT missions. You have Sentinel-5, which is mainly there to collect data about the atmosphere. You have Sentinel-6 coming up. But you also can see that the nice thing in this overview, you see that Sentinel-1 and Sentinel-2 are not just one satellite. Sentinel-1 are two satellites, or currently one satellite. One was taken out of commission. And Sentinel-2 are also two satellites, but they're already planning Sentinel-1C and Sentinel-2C. So every 5 to 6 years, additional satellites will be launched, which have the same specification as the current ones. So we will have a continuous long time series of the Sentinel-1 and Sentinel-2 missions. The same you can also do for the NASA-based or the US-based satellite missions out there. Most important of you knowing Landsat 7, but Landsat started already with Landsat 1 in 1970s. Currently, we're running everything on Landsat 8, and Landsat 9 was launched. Also here, you get a really nice long-term series of satellite data. And the same we also can do with JAXA, so the Japanese aerospace space agencies. They have also quite a lot of impressive satellite systems in space, monitoring our Earth around the clock. But as a take-home message, you can see on the right side, the data amounts we are getting out of satellite remote sensing are drastic. So you can see in 2013, so before the Sentinel missions came up, we were talking about roughly half a petabyte per year. Easy to handle. Starting with the Sentinel missions, we are coming up now with 4 to 5 petabyte on data per year, which is raw data. So we still have to process that. It's expected to go up to 2030 up to 10 to 12 petabyte per year. So these data amounts are not easy to handle, as let me say, you need really specialists in order to process all these data sets. You have interoperability issues, so you cannot take and just combine all the satellite data and hope that you get a product which is lasting over time, because each sensor has to be looked standalone, or you have to harmonize the sensors against each other. I will show also a slide later why. A lot of missions are only for research, so that's the reason, as you can see in this list, we have mainly Landsat, MODIS, and Sentinel satellites herein because they are long-term planned data sets. High-resolution long-term data sets are available since 1983 with the Landsat mission, but only dense temporal resolution, so with 5 daily revisit time or daily revisit time, that is possible since 2016. So I tried to condense all this information in a nice overview of the most important satellite systems out there. So if you look, the Landsat mission, as I already said, we have now already Landsat 1 to 8, or Landsat 9 is also planned. And you can see there's quite an overlap of some of these missions. Like, for example, Landsat 5 was going from 1984 to 2013, but already in 1999, Landsat 7 started. So you had there quite an overlap, 14 years of overlap of both satellites. So you can harmonize these quite nicely because we know the specifications. But you also see they are available in 30-meter resolution on a global scale with 16 days revisit time. So that's really most of, it's a 45 to 50-year archive of valuable data, global scale, but you have to analyze most of this data by yourself. There is some products coming out, but most of the data sets you have to run by yourself. So you have the MODIS satellite, which was launched in 1999, and in 2002 was the second. So MODIS itself is two satellites missions. That's the Aqua and the Terra satellites. Overall, that is not only one satellite missions. I think there was quite revised because they're also running already over 20 years. You have there quite a lot of bands, so you can manage to see a lot of the electromagnetic spectrum, but the resolution is 250 meter only for two bands, and some bands are provided in 500 meter spatial resolution. At most of the other bands are at 1 kilometer scale. You have the AVHRR mission, which is running since 1978. Overall, over the time, they launched 19 satellites for this mission, and but they are quite consistent, but only you get 6 bands and at a resolution of 1 kilometer. So there you can generate quite easy, long time series product at 1 kilometer resolution. But always the question is, that will help you maybe if you want to get an overview of a country, of European overview, but it will not help you quite a lot if you want to have if you have a small test site, then you will get nearly no information. Then we have the SPOT VGT PROBA-V Sentinel-3 archive. So SPOT VGT was launched in 1998. PROBA-V, which is a satellite which was launched by Vito, was launched in 2013 and replaced SPOT VGT. And the Sentinel-3 mission from ESA was launched, I forgot this, was launched, I think, in 2019, and that replaced now PROBA-V. So there you get also a nice overview over Europe at a 1 kilometer scale, but starting with PROBA-V. So we're starting with 2013, you get this information also in 100 meter resolution every 5 days and 300 meter every day. So if you want to generate long time series for Europe, that's quite a good data source. And starting with 2014, the Sentinel-1 mission started. That is radar data with a resolution of 5 to 40 meter, depending on the band you want to use. And starting with 2015, or more in detail, starting with 2016, really you get detailed Sentinel-2 data, which is with a resolution of 10 to 20 meter and a temporal resolution of 5 days. So that is the most recent data set you can use. So what I said already, you cannot use all satellites and just put them together and generate one product. As you can see here, that is a nice overview of a comparison of Landsat 7, Landsat 8, and the Sentinel satellites. And you can see that the bands, so what represents the frequency of the electromagnetic spectrum, are not always matching to each other. So they are overlapping and they are not always measuring the same area of the electromagnetic spectrum. So you have to harmonize these data sets. You have to make them comparable in order to use them. So there are not a lot of data sets out there which harmonize time series of satellite data set. One of the best available data set currently is the Landsat HLS-2 project, which is harmonizing Landsat 8 and Sentinel-2 data. So you get 30-meter resolution data starting with 2013 on a nearly 2 daily to 3 daily basis. And all the satellites which are within this mission, so overall in this case 3 satellites, and starting with Landsat 9 we have 4 satellites, are harmonized to each other, so you always get the same results. So for example, if you would calculate the NDVI from this data set, you can directly use this. If you would calculate the NDVI of each of the satellites independently, you could not even compare the NDVI from one satellite to another satellite mission. ESA is planning also a Sentinel-Landsat product at 10-meter resolution, so nearly the same archive where they will reprocess the Landsat 8 and Landsat 9 data into 10-meter resolution instead of 30-meter resolution. But other than that, there's nearly no harmonized products out there. You can generate them by yourself in portals like Google Earth Engine or OpenEO, but that means you have to do it by yourself. In the last years, we, as a remote sensing community, came up with a new standard which we call ARD, Analyzed Ready Data. So that is something which helps quite a lot in data processing because nearly all new and partly reprocessed archives are available as ARD standard. That means all the data sets are pre-processed to a part that a researcher can directly use them. So you don't have to deal with atmospheric correction, geometric correction, harmonization, BRDF effects, and all these other things you would have to deal with when normally you get the data for free when you download it. ARD data is mainly directly accessible in the Google Earth Engine and Planetary, as well as also in the CDSE, so in the OpenEO platform from Vito. It will save you up to 80% of your time in pre-processing, but the last 20%, so the real hard stuff, that you have to do still by yourself. So analyzing the data, making sure you're using it correctly, that is still up to you. So what is coming up in the next years? There are coming up quite a lot of missions from ESA as well as NASA dealing with problems or with challenges we saw over the last years, like biomass estimations, temperature measurements, as well as radar missions. So there are specific missions coming up for example, biomass. That's a biomass mission from NASA. There are several missions coming up in the thermal and optical band from ESA and CNSES. But all these data sets will be mainly not available within OBSESSION, but there are synthetic data sets available in case you want to test your algorithm with higher resolution data or with data sets which are will be available in the future. Call for action. What we want to do with that is, as you know, in Work Package 2, we're trying to help you with benchmarking and developing EBV algorithms, as well as providing remote sensing biodiversity products. But in order to do that, we have to know what the different work packages on OBSESSION need. So every use case should first think about which biodiversity variables or remote sensing products do you want to evaluate within your use case. So that would be the first part. Then you should think about, is an EBV algorithm already existing or defined for this EBV you want to run? So you can check that on the EBV portals you find here on slide 7 to 12. If there is an EBV already existing, then please download it from the portal, check if it's sufficient for your case, or if we have to adapt the algorithm, maybe you need it in a higher resolution and so on. If it's not existing, then you should check which are the existing remote sensing biodiversity products, so slide 13 and 14, or if there are existing methodologies to calculate. We will help you to benchmark these algorithms. That's part of the work package 2. But you have to have a look what biodiversity product you really need in which resolution and so on. If these remote sensing biodiversity products are not existing, then you can go through the remote sensing data time series to generate these products either by yourself. So you can have a look on slide 15 to 20 to check what are the remote sensing data you want to use or which one should be the best options for you. We can also help you there for the algorithm benchmarking and maybe development. But then the last point is you have to fill it into the data list so that we know what you need. Currently, this list only exists out of 9 entries, so not a lot of data sets which we should provide within OBSESSION. But you have now the chance, that's a living list, so you have now the chance to to think around, go again in and add missing data sets as well as when you need them. And then we can go through and check if we can supply this data set directly, or do we have to do more work? As an example, Wageningen requested, for example, Sentinel-2 10 to 20 meter composites of the vegetation season, or maybe even monthly composites. From a technical point of view, that is not a problem. We could roll them out really in the next days, but it's more than we have to talk about, do you really want them for whole Europe in one run? Because then we run out of budget within the first, I would say, 20 days. So that it's a little bit discussion there also needed as we can do today. So that was it also then from my part. And the slides will be anyhow provided to you by Maria later.